Hello guys, this is Dr. Hadi here. Welcome to the Medical Globe. Today's topic is exclusively on nucleic acid, and each and everything you want to know about nucleic acid. I brought a lot of questions about nucleic acid today, and we will discuss this. These are actually 14 questions regarding the nucleic acid. These 14 questions are, the first one is the introduction of nucleic acid. This is our start. And then structure of nitrogenous bases. Then how to memorize the structure of nitrogenous bases. Mostly students feel it difficult. They can't memorize the structure, a little bit difficult. And then the structure of nucleotide. Fifth question, difference between ribonucleotide and deoxyribonucleotide, difference between the DNA and RNA, structure of DNA and RNA. This is the main topic of today's lecture. And then some abbreviation used in the nucleic acid, then minor bases, and next question is how two nucleotides are linked together, and why we use what is this 5' and 3', 5 and 3 in the topic of nucleic acid, why there is 2 hydrogen bonds and 3 hydrogen bonds between the nitrogenous bases, especially 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine. Why? We will find the reason of this today. And then the function of nucleotides, and then some basic terms like template strand, antisense strand, and sense strand, complement word. These all will be discussed in today's lecture. I will take this page again at the end of the lecture, and we will check any question left. If it is, we will discuss it again. So let's start with the introduction of the nucleic acid. Nucleic acid, the first, its discovery. The name of this nucleic acid came from its discovery. Nucleic come from the word nucleus, right? This is from the nucleus, and acid is by nature. You know, compounds are either acidic or basic. For the first time when that compound was discovered, it was discovered in the nucleus. According to some books, in the nucleus of a pus cell, and according to some other books, in the nucleus of a sperm cell of the fish. So we are not concerned with the cell, but the main point is there that because it was discovered in the nucleus, so the word nucleic is there. Okay. Second, acid, because the compound was acidic by nature, not basic, it was acidic, so the name was nucleic acid. Okay, now we will start the definition of this nucleic acid. What is nucleic acid? Dear students, the lecture will be general in the beginning, right? Yeah, you may feel so easy, you will think that this is lower class level, but slowly and gradually we will go to the advanced level, because most of the students are average student and some are very sharp students, so we will have to carry on all these two type of students, whether they are average or genius students or smart students. So the definition of nucleic acid is that a nucleic acid is a polymer. A polymer, what is a polymer? When small molecules combine and they form one large molecule, that large molecule is called a polymer. Just as you can see, for the convenience, I have mentioned small circles are there. Look at that. These small circles are smaller units, right? These are the smaller units. So 1, 2, 3, 4, 5, 6, and so on. These smaller units, when combined together, then it will be one single molecule. This will be one single large molecule. This single large molecule will be called a polymer. a polymer. So the first thing is about the nucleic acid is that a nucleic acid is a polymer, right? And then every student wants to know that the smaller units which are combined, is there any name for these smaller units? I will say yes. These smaller units are the most important thing to understand the DNA. If we are able to understand these smaller units, I guarantee we will get everything about nucleic acid. So this is very special to know about these smaller units. So what is the name of these smaller units? The name of each unit is called as nucleotide, right? Means many nucleotides combine to form nucleic acid. Of course, we will discuss one single general nucleotide. Look, nucleotide is of two types. So before we discuss the two types of nucleotide, first we will discuss the general nucleotide. What is a nucleotide? Let me clear this idea to you people, and then we will come to the types of nucleotide. So each nucleotide consists of three parts. The first one is the nitrogenous base, the second one is pentose sugar, and the third one is phosphate group. These three compounds, you say, are three units, you say, these three combine to give us one nucleotide. Okay, let's start. What is nitrogenous base? A nitrogenous base is a compound which contain nitrogen atom. Definitely there is word nitrogen, and that nitrogen is capable of giving its lone pair. So this will make the compound basic. So it is called as nitrogenous base. You know, a base is a compound that can give its electron pair to other. So a nitrogenous base is also a compound which contains nitrogen, and that nitrogen is capable of giving its lone pair. You will be given the complete structure of the nitrogenous base just on my right side. You will have to wait for this. First, simple. A nitrogenous base is of two types. One is single ring and other is double ring. A single ring can be called as pyrimidine. We call it pyrimidine, right? Single ring, pyrimidine, and the double ring can be called as purine, right? So the double ring can be called as purine and single ring pyrimidine. So it means purine and pyrimidine are nothing else but these two are nitrogenous bases. If pyrimidine is a nitrogenous base, it must have nitrogen atom. So for your convenience, I am not going to write the complete structure. The complete structure will be on my right side, but just the simple idea will be that this is a ring, right? And here we can write one nitrogen here at that position and other nitrogen at this position. The remaining corners, you know, all are 6 corners, but the remaining corner will be of carbon atom, right? So every corner here will be of carbon, right? Carbon, and these two will be nitrogen. So here an average student can understand that this is a nitrogenous base because this compound contains nitrogen, although this nitrogenous base is in the form of a ring rather than in a straight form, an open structure, but it is a ring. Remember, this is a ring form of nitrogenous base. We have 2 nitrogen here. Similarly, this is also a nitrogenous base. So for your convenience, again I have to write 2 nitrogen here, right? So double ring and 2 nitrogen here, right? So these are double rings. So 1 ring and 2 ring. This ring is just like pyrimidine. 6 corners are there. But with this ring, we have another 5 corner ring, right? So one 5 corner ring, one 6 corner ring, both these combined, it will give us purine. Okay, so this is a nitrogenous base. Pyrimidine is the name given to the 3 compounds. These 3 compounds are cytosine, thymine and uracil. Cytosine can be written as C, thymine can be written as T in short form, and uracil can be written as U. Remember, these 3, cytosine, thymine, uracil, these 3 are pyrimidine nitrogenous bases. And purine are also two types. One is guanine, written as G. Another one is adenine, we write it as A. So guanine and adenine, these 2 are also nitrogenous bases, and these 2 are collectively called as purine. So the structure of guanine and adenine will be delivered to you with full structure on the right side. Now, this was the first part of just one nucleotide. As I told you that one nucleotide consists of 3 parts. The first part is just finished here, and the second part is pentose sugar. A pentose sugar it consists of 5 carbon, right? And there is 5 corners, right? This is the first corner, it contains carbon. This is another carbon, it also contains carbon. This is the third corner, this is the fourth corner, and the 5th corner does not contain carbon, it contains oxygen. The 5th carbon, you will find it here, right? So 5 carbon, 1 oxygen, this is a pentose sugar. Of course, we cannot discuss the sugar in more detail because this is the part of carbohydrate. We will discuss this carbohydrate in the upcoming lectures, but for simplicity, this is just a sugar, and inside sugar this is pentose sugar. You can write HOH on upside and downside, right? HOH, 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 but no need to write these H and OH in today's lecture, right? I think I can write it. Okay. The pentose sugar is of 2 types. You must remember that. It will be useful for you and us in next time. The pentose sugar is of 2 types. One is ribose sugar, the second one is deoxyribose sugar. Ribose sugar is that sugar which contain OH on carbon number 2. This is always carbon number 1 in case of pentose sugar. In book of Satyanarayan and in the book of Leninger, you can find prime on the numbering, prime on number 1, and it is carbon number 2, you will find prime on 2, right? So it is actually, we have nitrogenous bases also, and we have sugar as well. The numbering can be done in the nitrogenous bases as well as in the sugar. So the numbering which is done in the nitrogenous bases are just simply 1, 2, 3, 4, 5, but the numbering that we use in the sugar will be given prime just to differentiate between these 2. So carbon number 1, 2, carbon number 3, carbon number 4, and this is carbon number 5. Only and only come to carbon number 2, 8 carbon number 2, if the oxygen is present, right? If the oxygen is present along with hydrogen, this will be called as ribose sugar, right? It will be useful for us when we discuss DNA and RNA, comparison of DNA and RNA. It will be very useful for us. So you have to keep in mind that what is ribose sugar and what is deoxyribose sugar. A deoxyribose sugar is that sugar which does not contain oxygen at carbon number 2. It contains only hydrogen. So that is why deoxy without oxygen, deoxyribose. It is ribose, it is deoxyribose. The third part is the phosphate group. A phosphate group is usually, we just write it like this, PO4, very simply. But the actual structure of phosphate, you will find it a single phosphate atom. Around the phosphate there will be 4 oxygen atom. Any one of the oxygen, anyone, right? Anyone, is no need that specifically, especially you always write double bond with this oxygen. Any one of the oxygen will be double bonded, and the remaining 3 oxygen you will find as a single bonded with negative charge. This phosphate is actually came from phosphoric acid, H3PO4, phosphoric acid, right? So keep in mind, that was the reason that we give the name acid to the nucleic acid. So sometimes we say nucleic acid because of this nature. So where these 3 PO4 is there, where the 3 hydrogen, actually the 3 hydrogens atoms are attached to these 3 oxygen. So when these hydrogen are removed, because it is acid has a tendency to remove its hydrogen, and when the hydrogen is removed in the form of proton, that oxygen will become negatively charged. So these 3 oxygen has just removed their hydrogen, so we write them as it is. Now we got the 3 main parts of one nucleotide, and now I would like to write just one single nucleotide here on. So I'm going to erase it. I'm just going to erase it. Okay, so this is. Okay. Now it is time to write one nucleotide, just one nucleotide. Okay, so first of all, the very first, I'm going to write the sugar first. Yes, this is the sugar, right? Okay, this is the sugar. And don't worry, you can just, some student can just write it like this, okay, straight. It's okay, fine, no problem. So this is just sugar. I'm going to write S here. Don't worry, it may be ribose or deoxyribose, we are not concerned with this now. If this is carbon number 1, at carbon number 1 you will find there will be a nitrogenous base, because there are 2 types of nitrogenous base and a total of 5 different nitrogenous bases. So any one among those 5 nitrogenous bases can be attached here. So I just write NB, nitrogenous base. It may be adenine, it may be guanine, it may be cytosine or thymine, and it may be uracil, anyone. We know that this is 4th carbon, and here we have the 5th carbon. At 5th carbon you will find PO4 phosphate group. So when a nitrogenous base is attached with sugar and there is no phosphate, we will call it nucleoside, right? Nucleoside. But when a phosphate is attached, it will be called as nucleotide, right? So girls and guys, now we got one nucleotide. This is the structure of one nucleotide. And come to these circles now. If I consider each circle represent this nucleotide, then we will be having such kind of a lot of nucleotides arranged, linked with one another, and we will call them a polymer, a polynucleotide, which is called as nucleic acid. Okay, furthermore, the nucleotide, as I told you, nucleotide will be of different types. Now we will come a little bit from general lecture to a little bit specific. Now we will specific now. Okay, the nitrogenous base can be any one. Like for example, if I just write here in place of nitrogenous base, if I write here there is adenine, right? So if I just attach it carbon number adenine, so it will become adenine nucleotide, right? And if I just write G here, it will be called as guanine nucleotide, right? So the whole name of the nucleotide will be given according to the nitrogenous base attached. These two things you will find in every nucleotide, but it will determine whether it is which nucleotide. And if you attach C, it will be cytosine nucleotide or cytidine nucleotide, whatever. So it means we can have 5 different nucleotides on the basis of the nitrogenous base. Because we have 5 different nitrogenous bases, so we could have 5 different nucleotides. This is first. Now, the structure in front of you is just a nucleotide. A nucleotide is of 2 types. One is called ribose nucleotide, other one is called as deoxyribose nucleotide. As you saw, ribose sugar and deoxyribose sugar. On the basis of sugar, we will give another name to the nucleotide, because you know sugar is of 2 types. One is ribose sugar, other one is deoxyribose sugar. Look, if I just attach here OH at carbon number 2, this will be called as ribonucleotide. That's simple. If there is no oxygen, only hydrogen is there, so we will call it deoxyribonucleotide. Deoxyribonucleotide. So this was another naming system for the nucleotide. So nucleotide can be named on the basis of the sugar or on the basis of the nitrogenous base. Keep in mind all these things. Now, dear students, once we got the types of nucleotide, and now I will take you people to the structure of all 5 types of nucleotide, different nucleotide structure. Please give me some time so that I can fix camera, your eyes on my right side. Okay, so this is now the structure of different nitrogenous bases. You just learned that nitrogenous bases are pyrimidine and purine. So here, once again for you people, we are discussing nitrogenous base, but in a little bit detail, because it makes the difference from the higher level and the lower level class. A nitrogenous base is of 2 types, as you saw: double ring purine, single ring pyrimidine, right? Purine may be adenine or guanine, and pyrimidine may be cytosine, thymine or uracil. If you want to get the structure of these bases, so here I have just mentioned something for you people. Adenine and guanine are double ring, so here is the double ring. Cytosine, thymine, uracil are single ring, so here is the single ring. Now, the first thing we must at least know this simple diagram. of double ring, this simple diagram and that one. Right, draw two rings. One is 6 corner ring and that is 5 corner ring. 2 nitrogen at these two position and 2 nitrogen and these two position. Here you can again write this kind of sort of diagram. You can write 2 nitrogen in this one. Now numbering is most important. We must give some numbering like 1, 2, 3 in order to understand the structure, to memorize the structure. For convenience, for simplicity, the numbering system of the double ring will start number 1 from this nitrogen, right? Nitrogen position. So 1 is not necessarily for carbon. 1 may be for nitrogen. Okay. So 1. Then come down 2, 3. Don't go here 1, 2, 3, 4. No, you must come here to this nitrogen. 1, 2, 3, 4, 5, 6. So 6 positions are there in that ring. And then 7, 8, 9. You can go either 7, 8, 9 or 7, 8, no problem. But this is most important, right? 1, 2, 3, 4, 5, 6, 7. And here you can start nitrogen from the nitrogen at the bottom, at the base. This nitrogen is very important. So you will start numbering with this nitrogen 1, 2, 3, and of course 4, 5, and 6. Now you will get these structures very easily by looking at what I have mentioned here with adenine. Look, this structure is both for adenine and guanine, but in case of adenine you will have to go to the carbon number 6 only. At the carbon number 6 there is NH2. So where we have 1, 2, 3, 4, 5, 6. Yeah, this is 6 carbon. So what you will do, you will just write an NH2 here at that carbon. This is fine. This is just finished. So that structure will become adenine, and you got this. You memorize it very easy now. If you want the structure of guanine, so you have to see that in guanine we need 2 carbon. One is the carbon number 2 and the other is the carbon number 6. At carbon number 2 there is NH2. So you must write here. Okay, I'm going to just erase it. At carbon number 2 we have NH2, and at carbon number 6 we have double bond O, right? Where is this one? Is carbon number 6. So here we have double bond O. So a student needs to memorize this only, and he or she can memorize the whole structure. And in this way we got the structure of adenine and guanine. Now, so this is guanine, right? So this is guanine. Okay. Cytosine C2, C4. It's C2. This is C2. We have double bond O, so this is called carbonyl group, right? This is carbonyl group. At the C2 position, remember those corners on which nitrogen is not present, all those corners means that there is carbon. Okay, so there is carbon. So at carbon C double bond O here. We usually don't write C, because everyone will understand the corner shows carbon. If it is not carbon, if it is something else, sulfur or nitrogen, then we must mention them. Otherwise, if it is carbon, there is no need to mention it. Okay, so at C2 we have double bond O, and at C4 we have NH2. So here we will write NH2. Now look, that whole structure now become what? Cytosine. But remember, once you attach double bond O here, then there is a slight problem of valency. Like this is carbon, so carbon has 1 bond, 2, 3, 4, 5 bonds. If here double bond O comes, then you will have to erase this bond, and then it become like it will be then easy. Then you can bring hydrogen here, right? So nitrogen will become it's 3 valency. Nitrogen must have 1, 2, 3 valency. Carbon must have 4 valency, right? Okay. if you want to write thymine, so I think now you can write thymine. Okay, at C2 you will write double bond O, fine. At C4 you will write double bond O here, double bond O. Remember, again, this is a valency problem. You can make it correct by erasing that bond. So, and at C5 we have CH3 here. We got CH3. Now this is the structure of thymine. Okay, and for thymine, C2, C4, C5 is there. And for uracil, uracil structure is just like the thymine. You just minus it like C5. If C5 is not there, uracil looks like thymine. So you just remove this C5, thymine become uracil. So if you know the structure of thymine, uracil is very easy. I hope, dear students, in this way you got the structure of pyrimidine and purine. And now here comes the most important question that usually students seek at the master level, that why there is hydrogen bond, two hydrogen bonds between the adenine and thymine, and 3 hydrogen bond between the guanine and cytosine. Actually, when we proceed our lecture further, when I discuss DNA with you people, then in the topic of DNA you will find that one nitrogenous base is attached with another nitrogenous base in just coming lecture, right? So usually purine is bounded with pyrimidine. I should not say usually, but all time, right? All the time purine is linked with pyrimidine. How? Just like in this way, adenine is always linked with thymine. You know, adenine is purine and this is pyrimidine. Guanine is always linked with the cytosine. Guanine is always linked with the cytosine. You just don't worry about uracil. Uracil actually comes in place of thymine, so it will be we will find it in the RNA, not in the, so we will keep uracil to be discussed in the RNA, not in the DNA. And there is 3 hydrogen bonds between the G and C and 2 hydrogen bonds between the A and T. I would like to discuss that. First, student sometime smart students, genius students, they usually think why there is 3 hydrogen bond between G and C and 2 hydrogen bond between A and T. The simple answer to them is that it is because of the structure. In the structure of guanine, let's suppose this is the structure of guanine, right? And this is the structure of cytosine we have to draw here, so, right? This is cytosine. Okay, come, we will make it. At C2 we have double bond O, at C4 we have NH2. So here we have NH2, right? Now I will make bond between these two, and I will tell you that why there is 3 hydrogen bonds. Okay, so this time I'm going to erase it because you can reverse the video, you can watch it again anytime. Okay, so this is a cytosine. Now if I just bring it a little bit closer, we will write it like this. Okay, okay, this is pyrimidine ring. We have a nitrogen here, we have a double bond O here, we have nitrogen here, and we have again nitrogen, but the H2 will be shown as like this. This is one H and this is another H, right? NH2. Here is nitrogen, right? Okay, and this nitrogen will be again shown as right, and H, right? So if we just see here, you can find that this one is the first hydrogen bond that can be made between these two oxygen. This is oxygen, okay, and got hydrogen, right? This is the first hydrogen bond. The second hydrogen bond will be okay, this is NH, and sorry dear, here we have to write one more hydrogen here. So when double bond O comes here, then the valency of carbon will become 1, 2, 3, 4, 5. So here this bond must be removed. It's fine now, 4 valency, but this nitrogen has got 2 valency. It means this nitrogen must have a hydrogen now, right? So hydrogen bond can also be formed between the hydrogen and the nitrogen as well, right? It is not usually formed between oxygen and hydrogen, between the nitrogen and hydrogen as well, because this is slightly positive and this is slightly negative in comparison with hydrogen. So there will be another hydrogen bond between that hydrogen and this nitrogen, right? This is slightly negative, this is highly electronegative as compared to the hydrogen. So the hydrogen bond is always formed between the partial charge hydrogen atom and slightly or more electronegative oxygen or nitrogen atom, right? So we got nitrogen here, another hydrogen bond will be there, and the third one, I would like you people to decide where the third hydrogen bond will be. Yes, it is here, right, between that oxygen, because it is electronegative and this is electropositive. So here another bond will be formed, another bond. So this is now the third. 3 hydrogen bonds are there. In case of adenine and thymine, the structure looks like same, but one of the bond will not be, means such kind of 2 bond will be there, the third bond will not be there, although you will find double bond oxygen would there and hydrogen also there, but the main problem is the distance. In case of adenine and thymine, the hydrogen and oxygen are a little bit away from them, the distance is too large, so the possibility of the third hydrogen bond is not there. That is why in adenine and thymine there will be 2 bonds, but in guanine and cytosine there will be 3 bonds, and that is the reason there's 3 and 2 bonds. Okay, this was the first. Second question is why always purine is bonded with pyrimidine? Why pyrimidine and purine, why they don't link with each other? I got 2 reasons from the book of Satyanarayan, that if we attach purine with purine, there will be problem of a space problem. And if we attach pyrimidine with pyrimidine, then according to that book of Satyanarayan, there will be a problem of distance. If we attach pyrimidine with pyrimidine, it will not be possible, because the hydrogen bond and the other atom that are going to make hydrogen bond will be too far away with each other, and that is why there will be no possibility of the pyrimidine bonding, and purine will not attach. There is a space problem. According to that book, they said that there is a specific space, right, in the DNA. In the DNA, if you bring purine P, only the space is only that kind of space that it can attach a pyrimidine You cannot bring another purine here because the space is limited, available space is limited. So I hope you understand that these two reasons are being shown in the book of Satya Narayan, that why it is purine cannot attach and pyrimidine cannot attach. In reality, in the real structure of DNA, purine is always attached with pyrimidine. Further specifically, purine is adenine in guanine, pyrimidine is cytosine in thymine. A is always attached with the thymine and C is always attached with the guanine. We will discuss this in the structure of DNA as well, but these were few points that I wish I could discuss with you, because most of the students at the master level, they want to discuss, they want to know the answer. So now we got the nitrogenous bases, and now I will erase all this once again, and now I'll come to the structure of the DNA and RNA. So as I told you in the beginning of my class, that we will first start simple lecture and then slowly we'll come in detail. So here is the nucleic acid. Once again, nucleic acid is of two types. The nucleic acid is of two types. One is the DNA, and the other one is the RNA. The DNA can be written as deoxyribonucleic acid, while RNA can be shown as the ribonucleic acid. Both the DNA and RNA, you got the structure in advance. Slight modification is there that I will tell you now. Whether it is DNA or RNA, both are polymers. So both are polymers, right? Both are polymers. That is also polymer. So polymer. Okay, sorry. Smaller units, nucleotide are there, but here also smaller units, nucleotides are there. The difference is that each unit here is called as a ribonucleotide. Ribonucleotide. So RNA is a polymer of ribonucleotide. And you know very well what is a ribonucleotide, because we just discussed this. Here every small unit will be called as deoxyribonucleotide. So deoxyribonucleotide, many deoxyribonucleotide combined, they will give us a polymer called the DNA. This is the first difference. Second difference, RNA is just single-stranded. RNA is just single-stranded. You can see only one single polymer, okay? But the DNA is double-stranded. It is double-stranded. Here, double-stranded means this is one strand, and here another strand. So two-stranded. If these are two-stranded, they must be attached with one another. You just don't worry. This is not the structure I'm going to tell you about the DNA. We will discuss the complete structure of the DNA as well. But it is just I'm going to give you a slight idea. So there will be hydrogen bond between these two chain. So double-stranded DNA, single-stranded RNA. Remember, what is the most common thing in between these two is that both consist of smaller units. It is nucleotide, more specifically ribonucleotide. These are also nucleotide, but more specifically they are called as deoxyribonucleotide. And I hope you will know that you got the whole idea about deoxyribonucleotide and ribonucleotide. Okay, now the DNA contain If it is deoxyribonucleotide, deoxy, definitely it means DNA contain deoxyribose sugar. And RNA, because it contains ribonucleotide, so definitely it contains ribose sugar. So these are the differences, comparison between the DNA and RNA. And the DNA carries biological information, genetic information, biological informations. Sometimes we call them as genetic informations. What is genetic information and biological informations? What you can see inside your body, your eyes, eye color, hair, skin, cells, inside the cell, each and everything is the biological information. And you know, when offspring are produced, right, a baby when is formed in the womb of mother, a baby gets all these structure just like his parents. Hand, two hands, nose at the same position, eyes at the same position. There is no mistake occur in the baby. Each and everything of the human body is transferred in that baby. So it means our body contains some sort of information, just like your USB, a memory card, a USB that used to store that used to store documents, right? You can save documents, you can save movies or songs in a USB. So USB carry these information in the form of data. Our USB, which contain millions of informations, are inside the DNA. So whenever the DNA transferred to the offspring, the DNA carries all the information to the offspring. So that's why we say DNA contain the heredity information, especially in human. But some viruses, they have heredity information in the form of RNA. So in human, we have the DNA that carries the biological information in the form of gene. I may give you some idea about the gene as well, because when we got the structure of DNA, we will get the structure of gene as well. So RNA is just get messages from the DNA. RNA works under the DNA. The DNA gives command, RNA do work, right? Especially just like in protein synthesis, the DNA give a message in the form of RNA, because we say DNA contain messages, DNA contain information. So DNA will not go there, DNA will just give its information. Whenever DNA gives its information, that is always in the form of RNA. RNA is always formed from the DNA, right? So RNA, with the help of RNA, you will get protein. And that protein which is made in our body come from the DNA. The information of the protein is present in the DNA. The DNA itself does not come. DNA gives the information in the form of RNA. So I hope you got this. RNA is of three types. RNA is of three types, but DNA is of single type, right? So three types of RNA are the three types of RNA. I will just write the name. I think today there is no need to explain the three types of RNA. One is called the mRNA, messenger RNA. The second one is called as the tRNA, transfer RNA, and the ribosomal RNA. Messenger RNA carries message, and transfer RNA carries amino acid. This is not the part of your topic today. This is not your topic, right? Ribosomal RNA carries, it is the part of ribosome. It is a part of ribosome. We will discuss this in the topic of protein synthesis. Let this topic come, right? Okay, so we got the difference between the DNA and the RNA. Now the structure of the the DNA. Okay, complete structure. I think the students were waiting for this, that which time the structure of the DNA will come. So this is the time that the structure of DNA will be discussed. Usually we write 2 lines like this, right? That is the DNA. Usually we write this. We write 5 here, and at the end of this we write 3 prime. Here we write 3 prime, and at the end of this we write 5 prime. Every students when see such kind of diagram, they come to know this is DNA, right? Okay, but if we go into detail, DNA consists of 2 strands. We just got this, right? DNA consists of 2 strands. Every strand consists of smaller units, right? Every strand consists of smaller units, and every unit is called as deoxyribonucleotide. I think we just discussed this, deoxyribonucleotide. So each deoxyribonucleotide is linked with another deoxyribonucleotide, and link hydrogen bond, right? So this looks like a ladder, right? A ladder. So the DNA structure resembles the ladder, especially the one which is twisted like this. So who were the scientists? Actually these were the Watson and Crick. These were 2 scientists gave the structure of the DNA, and nowadays the DNA structure has different forms from A, B, C. There are 6 different structures of the DNA discovered yet. Among these 6, 3 are common: A form, B form, and Z form. A, B, and C. Among these 3, the most important one is the B form. The B form structure was discovered by the Watson and Crick, and here is what we are discussing, dear students. Now each circle is represented as 1 deoxyribonucleotide. So I'm going to write the first deoxyribonucleotide, and you will get the answer how the 2 nucleotides are linked. So here we have the first nucleotide, right? This is the first nucleotide. This is sugar, and here we have a nitrogenous base. Any one of your choice. I will write here A. A is enough, right? There is no need to write the structure of the adenine again. No, you got the structure. Now we just write A, and here you will write a phosphate. Here we can have another sugar. Here we have another sugar. Okay, I think it is a little bit at the bottom. So here, okay, is another sugar. So I can write here another nitrogenous base, guanine. Here cytosine. Remember, always you must attach this at carbon number 1, and this is phosphate here. Now the main thing is that how one nucleotide is linked with another nucleotide. This is carbon number 1, carbon number 2, carbon number 3. At carbon number 3 we have OH, right? Everyone knows very well that at carbon number 3 we have OH, and here again OH, right? Carbon number 3. Do you know, I hope you must know, ester bond. What is this? When an acid combines with alcohol, then what is formed? A new compound is formed called as ester, and the bond is called as ester bond. Yes. Alcohol will be designated as COH mostly, at carbon OH that is alcohol. An acid, just like any acid, like phosphoric acid. So phosphoric acid here, like we've just discussed the structure of phosphoric acid. This is C double bond O, OH and OH and again OH, right? You got this, right? So this is phosphoric acid, and here we have alcoholic OH, right? This is sugar, but the OH is considered as alcoholic. Now a bond will be formed between the phosphate and the OH, right? What will happen? The OH, the phosphate will give its OH, right? Here is P, make a bond with O. Here I'll just write here. This is a phosphate. If I write it in this form, it will be very good, I think. Here, okay, yes, P double bond O minus, right? Another O, here we write CH2 and a bond. Okay, if I just don't write it, so we will just write it as O. Okay, here a new bond is formed between the CH2 and the phosphate. This bond is called an ester bond, because in this bond the alcoholic part means OH has participated, and from the phosphate side again OH has participated. The OH from the phosphate, remember, the OH from the phosphate and the hydrogen from the alcoholic group, these two will combine to form H2O. So here one water molecule will be released. Remember, the OH will be released from the phosphate and the hydrogen will be released from the OH. Remember, there is OH on carbon number 5. And remember, there is OH on carbon number 5. It's C5. We have actually CH2OH, right? So here is CH2OH, right? Here is CH2OH. Here is again CH2OH. Here is again CH2OH. There is no need to show CH2, but OH is important. So whenever a phosphate comes here, the phosphate will make a bond with that OH. So hydrogen will be removed from the OH while the phosphate will give its OH. So one OH from the phosphate and a hydrogen from the OH group, these two will combine in the form of H2O. So one H2O molecule will be released, and finally we will be having a bond between the phosphate and the C5. This is called as ester bond, right? Ester bond. Now similar is the case with another OH. Look at that. Here at carbon number 3 again we have OH group. So of course the phosphate have OH group as well. So once again this phosphate will give its OH and the hydrogen will come from this place. So this hydrogen will be removed and OH from the P will be removed, and one more H2O molecule will be released, and there will be a bond between that phosphate and this oxygen. So if I just change the color, means this is the oxygen which is the part of the ring CH2OH, and this is the oxygen which is the part of the ring CH2OH. So this is one ester bond and this is another ester bond, and there is a phosphate between the two nucleotide. Position number 3 upside and position number 5 downside, one phosphate is linked. So therefore the name of this bond is, maybe the students who are watching, they will tell me that this is what, I can't hear you, but I hope you will say this is called as di-ester bond, right? Okay, I can just repeat it, that here is OH and the phosphate will make a bond with that OH. Whenever it make a bond, the phosphate will give its OH, and the hydrogen from the OH will be combined in the form of H2O and a bond will form. This oxygen is not the oxygen of the phosphate, although we know that phosphate also have oxygen, but this oxygen is of the ring, and the phosphate's oxygen has been removed in the water. Similarly the case here. So I think now there is no need to write it again and again. So now we will just write here PO4, and directly we will link like this, right? Okay, so this is one strand of the DNA, one strand of the DNA. This is just one strand. Now we have another strand here, but if you want to know another strand, you must write it like this. O is at the downside, right? This is one sugar, but this time the O must be at the downside. And again we have one more sugar here, and again we have one more sugar here. Okay, yes. This time that is carbon number 1, 2, 3, 4, and this is carbon number 5. 1, 2, 3, 4, and this is carbon number 5. 1, 2, 3, 4, and this is carbon number 5. Okay, just like here between 3 and 5 we got phosphate, similarly here between 3 and 5 we will got a phosphate PO4. Here 3 and 5 we will get a phosphate PO4. So in this way we got another strand. These are the two strands, right? And at carbon number 1 of the sugar we write nitrogenous base. So if it is nitrogenous base A, there must be thymine. This is called as base pairing specific. Adenine is always linked with thymine, as I told you. And in this time here, if it is guanine, so there must be cytosine, and if it is cytosine, then there must be guanine. Okay, so this is one deoxyribonucleotide chain, and this is another chain. These two chains must be linked, and so there is hydrogen bond that makes the nitrogenous base link with each other by the help of hydrogen bond. Adenine is purine and thymine is pyrimidine. So there will be 2 hydrogen bond and there will be 3 hydrogen bond, right? We have already discussed in detail structure wise, so there is now no need to put the structure again. And again here 3 hydrogen bonds. One thing more, if it is 1, 2, 3, 4, this is carbon number 5, and at the end you can see this is carbon number 3. So whenever we write the DNA in simple form, we just write one line here. If it is 5, so here we write 5 prime, and at the end of this we have 3 carbon and we write as 3 prime. Right? Here we write 3 prime. And the second one, look at that. Here is the third carbon, right? So we will write, this is the third carbon, and at the end we have 5 carbons, so we write it 5 prime. This is called as anti-parallel, right? This is called as anti-parallel. You can write it here also. If I present this one chain in the form of one line, so it is 5 and 3, that is 3 prime and 5 prime. Now in more simple way, we write the DNA structure like this, right? This is a little bit more advanced structure of the DNA. Here you can see grooves are there. Look at this, a small groove and there is a large groove, right? It's a large groove. Then also we have small groove and then we have large groove. The smaller one is called as a minor groove, and the larger one is called as the major groove, right? The major groove, the minor groove. The DNA has its diameter. If you just check the DNA here, the diameter is 2 nanometer diameter. One more thing, 3.4 nanometer. This is one spiral, right? One rotation. Between that place and this place, the distance is there. That is 3.4 nanometer is the distance between the two rotations, right? And there are 10 base pairs. How many base pairs between one rotation? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pairs are present in each turn. Turn is more good word to use. This is one turn, 3.4 nanometer, 10 base pairs in each turn. This is B form, B structure of the DNA. In A there is 11, and in Z there are 13 base pairs, but we are not concerned with this. The structure that was given by the Watson and Crick contain 10 base pairs in each turn. So 10 base pair, and that is the length. The second one is what is the distance between that base pair and this base pair, between the two base pair, right? Actually I can write it here. If we say DNA is like this, right? So what the distance between the base pair is 0.34 nanometer. 0.34 nanometer is the distance between the base pairs. And one thing more, sugar phosphate. If we consider the DNA as a ladder, so the sugar phosphate will make that sprung, right? That another sugar phosphate will make this side, while the steps, yes, on which we just place our steps, the steps is made of the nitrogenous bases. So nitrogenous bases will make the steps, while the two rails, I think rail is more good word to say, the two rails are being made by the sugar phosphate. If you want to know the structure of RNA, just raise this side completely. The single strand that left will be called as the RNA. But one thing keep in mind, in the DNA you will find adenine, guanine, cytosine and thymine. But in case of RNA you will find adenine, guanine, cytosine, but in place of thymine you will find uracil. Keep in mind, this is the only difference between the next difference between the DNA and RNA, that in place of thymine you will find uracil. You will never find thymine in the RNA. So okay, some abbreviations were there, like here I just wrote some abbreviations. Yes, the abbreviations are, here we have some abbreviations like DAMP and DGMP and DCMP. D means deoxy adenine monophosphate. If we have a sugar, right, and we attach one phosphate here and we attach here adenine, so this is adenine nucleotide. Now there is a question, if this nucleotide is the part of the DNA, because it can come in the RNA as well, if this can come in the DNA, if this is the part of DNA, so we will write it DAMP, adenosine monophosphate, one phosphate. D is for DNA. And if it is the part of the RNA, so there is no need to write D. Then every student will understand AMP means this nucleotide is present in the RNA. So DD, whenever you find small d, it means that these nucleotides are the part of the DNA. DGMP, guanosine monophosphate, cytosine monophosphate. Okay, I hope you got this, dear students. Now finally we got all these questions. I think almost we got all these questions. If still one thing, minor bases, what is minor bases? One was nitrogenous base like purine and pyrimidine, right? The minor bases are These are actually the purine pyrimidine. Just don't worry. If you have pyrimidine, let's suppose this is pyrimidine, okay? Right. There is, suppose here is nitrogen, and if I just modify it, a slight change, I bring small change here. If I bring here methyl group, okay, the methyl group on this nitrogen was not present in the nitrogenous bases we discussed, but this is a kind of new nitrogenous base that contain methyl group, so we call it methylated nitrogenous compound. Such modified nitrogenous bases are called as minor bases. Minor means they are not present in large quantities in the DNA and RNA, but they are present in a very small amount. These are present in the human, in the DNA of the human, in viruses, and even in plants. What is the function of the minor bases? Two functions. They protect the informations and they regulate it. Our DNA contain informations. Yeah, there are a lot of information are there. These information needs to be regulated as well as protection must be there. So who provide them the protection and the regulation? That is the another type of bases called as the minor bases. So whenever you find these minor bases, you will get the idea. I hope students finally you got my whole lecture. Sorry it was so lengthy, but the topic was like this. I hope you got this. Please remember in your prayers, always have a thumb up if you like it, and as usual, smile must be there on your face because it is both healthy for you and your family. Thank you. Bye bye.